Microcapsule applications in regenerative medicine represent one of the most exciting intersections of engineering and biology, where tiny engineered structures are used to support the healing and regeneration of damaged tissues. At their most basic, microcapsules are small, spherical constructs that consist of a core, often loaded with cells, drugs, or growth factors, encapsulated by a semi-permeable shell made of biocompatible materials such as alginate, collagen, or synthetic polymers. This shell is designed to protect the encapsulated contents from the host's immune system and to control their release over time. Imagine microcapsules as tiny self-contained factories or delivery vehicles that carry healing agents directly to the site of injury, much like a carefully programmed drone delivering medical supplies to a remote location. One of the core principles behind using microcapsules in regenerative medicine is the ability to achieve localized and sustained release of therapeutic agents. For instance, in cases such as osteoarthritis, where cartilage degeneration leads to joint pain and dysfunction, traditional treatments might involve systemic administration of drugs that can cause unwanted side effects and may not reach the target tissue in sufficient concentrations. In contrast, microcapsules can be engineered to encapsulate stem cells or bioactive molecules and then be injected directly into the joint. The semi-permeable shell of the microcapsule permits the diffusion of essential nutrients, oxygen, and therapeutic agents while shielding the encapsulated cells from immediate immune attack. This controlled release mimics the natural healing process, where growth factors are released gradually to stimulate tissue repair. You might ask, why is a sustained release important? The answer is that maintaining a therapeutic level of a drug or growth factor over an extended period is critical for effective tissue regeneration. A rapid burst release might provide an initial stimulus, but could quickly drop below effective levels, whereas a sustained release ensures continuous support for the healing process. The science behind microcapsule design involves careful consideration of material properties and engineering strategies. The shell's composition is chosen to be both biocompatible and biodegradable, ensuring that once the microcapsule has served its purpose, it will break down into non-toxic byproducts that can be absorbed or excreted by the body. For example, alginate, a naturally derived polymer from seaweed, is widely used because it forms gentle biocompatible gels that can be cross-linked with calcium ions. This cross-linking creates a semi-permeable membrane that controls the diffusion of molecules. In some advanced designs, scientists modify the shell with additional layers or incorporate degradable linkages that respond to environmental cues such as pH or enzymatic activity. A common question that arises is, how do these modifications affect the release rate? The answer lies in the shell's thickness, cross-linking density, and the chemical nature of the bonds. By tweaking these parameters, researchers can fine-tune the rate at which the encapsulated agents are released, much like adjusting the nozzle on a sprinkler system to control the water flow. Microcapsules also play a critical role in cell transplantation therapies, a cornerstone of regenerative medicine. When cells are transplanted directly into a patient, they are often vulnerable to the host's immune system, which may recognize them as foreign and mount an attack. Microcapsules act as immunoprotective barriers that shield these cells while still allowing essential exchange of nutrients, oxygen, and waste. A striking example of this is in the treatment of type 1 diabetes, where encapsulated pancreatic islet cells have been used to restore insulin production. The microcapsules protect the islet cells from immune destruction, while their semi-permeable membranes allow insulin to be secreted into the bloodstream and glucose to diffuse in, thereby maintaining normal blood sugar levels. You might wonder, is this approach effective in the long term? Research indicates that when optimized, encapsulated cell therapies can function for extended periods, providing a steady supply of therapeutic proteins while minimizing the need for systemic immunosuppressive drugs. Another fascinating application of microcapsule technology is in the area of tissue engineering scaffolds. In regenerative medicine, scaffolds provide a temporary structure that supports cell growth and tissue formation. Microcapsules can be incorporated into these scaffolds to create a microenvironment that mimics the natural extracellular matrix, ECM. The ECM not only provides mechanical support, but also presents biochemical signals that guide cell behavior, such as differentiation and migration. For example, in cardiac tissue regeneration following a heart attack, Microcapsules loaded with cardiomyocytes or progenitor cells can be integrated into a biodegradable scaffold. As the scaffold degrades over time, the microcapsules release their payload in a controlled manner, facilitating the gradual replacement of scar tissue with functional heart muscle. This process is similar to constructing a building, where temporary scaffolding is used during construction and then removed once the structure can support itself. Microcapsules are also being designed as smart delivery systems that respond to specific physiological triggers. These responsive microcapsules can change their permeability or degrade when exposed to certain conditions, such as an acidic environment, elevated temperature, or the presence of specific enzymes associated with inflammation or tissue damage. In the context of regenerative medicine, this means that a microcapsule could remain intact under normal conditions, but rapidly release its contents when it encounters the pathological environment of a wound or tumor. For example, in the case of an inflamed joint, the local pH may drop due to increased metabolic activity and inflammatory processes. A pH-sensitive microcapsule could be engineered to degrade more quickly under these conditions, ensuring that a higher dose of anti-inflammatory drugs is released precisely when and where it is needed. You might ask, how is the sensitivity of these microcapsules adjusted? The answer lies in the careful selection and modification of the polymer materials. By incorporating specific chemical groups that are stable at physiological pH but break down in acidic conditions, scientists can design microcapsules with tailored responses. Moreover, microcapsule technology in regenerative medicine is being advanced through the integration of real-time imaging and monitoring techniques. By labeling microcapsules with fluorescent markers or contrast agents, researchers can track their distribution, degradation, and release kinetics in vivo using imaging modalities such as MRI, PET, or optical imaging. This non-invasive tracking is invaluable for understanding how microcapsules behave once implanted, allowing for adjustments in design to improve their performance. For instance, by observing the gradual dispersion of a fluorescent signal from a cluster of microcapsules in an animal model, scientists can deduce the rate at which the capsules are degrading and releasing their therapeutic agents. This feedback is analogous to having a live telemetry system in a spacecraft, providing continuous data that can be used to adjust the mission parameters in real time. The versatility of microcapsule applications extends further into the field of regenerative medicine by enabling the co-delivery of multiple therapeutic agents. In many complex tissue repair scenarios, a single factor may not be sufficient to promote full regeneration. Microcapsules can be engineered to carry a combination of cells, growth factors, and other bioactive molecules, each with its own release profile. For example, in bone regeneration, a microcapsule might be designed to first release an angiogenic factor, such as VEGF, vascular endothelial growth factor, to promote the formation of new blood vessels, followed by the gradual release of osteogenic factors to stimulate bone formation. This sequential delivery is critical because successful tissue regeneration often requires a well-orchestrated sequence of biological events. You might ask, how do researchers achieve this sequential release? The answer lies in the layered design of the microcapsule, where each layer has distinct degradation properties or is sensitive to different environmental triggers, ensuring that the release of each factor occurs at the optimal time. Scaling up the production of microcapsules for clinical applications is another area where technological innovation is making a significant impact. Traditional methods of microcapsule fabrication, such as emulsification, often produce capsules with a wide size distribution and limited reproducibility. Recent advancements in microfluidic technologies now allow for the high-throughput generation of highly uniform microcapsules. In these systems, microfluidic channels and droplet generators are used to produce droplets of precise volumes that serve as templates for microcapsule formation. This approach is similar to using a state-of-the-art cookie cutter that produces perfectly uniform cookies every time, ensuring consistency across large batches, a crucial factor for meeting regulatory standards and ensuring predictable therapeutic outcomes. The potential of microcapsules in regenerative medicine is vast, and ongoing research is continually pushing the boundaries of what these tiny devices can achieve. From their role as immunoprotective barriers in cell transplantation to their function as smart, responsive delivery vehicles and integral components of tissue engineering scaffolds, microcapsules offer a multifaceted approach to tissue repair and regeneration. Their ability to provide localized, sustained, and controlled release of therapeutic agents not only improves the efficacy of treatments, but also minimizes systemic side effects, a major consideration in modern medicine. In conclusion, microcapsule applications in regenerative medicine are a testament to the power of combining engineering ingenuity with biological insight. These micro-scale systems are designed to mimic natural processes, providing controlled environments for cell growth, therapeutic delivery, and tissue regeneration. With innovations such as smart responsiveness, high-throughput microfluidic fabrication, and real-time imaging integration, microcapsules are poised to revolutionize regenerative therapies for a wide range of conditions. From joint degeneration and cardiac repair to neural regeneration and beyond, the journey from bench to bedside is being accelerated by these advances, offering hope for more effective, personalized treatments in the future.